Many carbohydrates found in nature are large polymers of glucose. Thus, a polysaccharide is a large polymer composed of many monosaccharide units, the monomers, joined in one or more chains. Homopolysaccharides are those composed of a single monosaccharide. Heteropolysaccharides are those made up of two or more different monosaccharides. Plants have the ability to use the energy of sunlight to produce monosaccharides, principally glucose, from carbon dioxide and water. Although sucrose is a major transport form of sugar in the plant, starch is the principal storage form in most plants. These plants store glucose in starch granules. Nearly all plant cells contain some starch granules, but in some seeds, such as corn, as much as 80% of the cell's dry weight is starch. Starch is a heterogeneous material composed of the glucose polymers amylose and amylopectin. Amylose, which accounts for about 20% of the starch of a plant cell, is a linear polymer of alpha-D-glucose molecules connected by glycosidic bonds between the carbon 1 of one glucose molecule and the carbon 4 of a second glucose molecule. The structure of amylose is shown here. Thus, the glucose units in amylose are joined by alpha-1,4 glycosidic bonds, shown here. A single chain can contain up to 4,000 glucose units. Amylose coils up into a helix that repeats every 6 glucose units. The structure is shown right here. Amylopectin is a highly branched amylose in which the branches are attached to the C6 hydroxyl groups by alpha-1,6 glycosidic bonds. The main chains consist of alpha-1,4 glycosidic bonds. Each branch contains 20 to 25 glucose units, and there are so many branches that the main chain can scarcely be distinguished. Glycogen is the major glucose storage molecule in animals. The structure of glycogen is similar to that of amylopectin. The main chain is linked by alpha-1,4 glycosidic bonds, and it has numerous alpha-1,6 glycosidic bonds which provide many branch points along the chain. Glycogen differs from amylopectin only by having more and shorter branches. Otherwise, the two molecules are virtually identical. Glycogen is stored in the liver and skeletal muscle. Glycogen synthesis and degradation in the liver are carefully regulated. As we will see later on in the course, these two processes are intimately involved in keeping blood glucose levels constant. And so this structure here shows both glycogen and amylopectin, and that they're both having these alpha-1,4 linkages and they have alpha-1,6 branching. The differences between the two is that the branches in glycogen are shorter and more frequent. The most abundant polysaccharide, indeed the most abundant organic molecule in the world, is cellulose, a polymer of beta-D-glucose units linked by beta-1,4 glycosidic bonds. A molecule of cellulose typically contains about 3,000 glucose units, but the largest known cellulose, produced by the alga Valonia, contains 26,000 glucose molecules. Cellulose is a structural component of the plant cell wall. The unbranched structure of the cellulose polymer and the beta-1,4 glycosidic linkages allow cellulose molecules to form long, straight chains of parallel cellulose molecules called fibrils. These fibrils are quite rigid and are held together tightly by hydrogen bonds. Thus, it is not surprising that cellulose is a cell wall structural element. In contrast to glycogen, amylose, and amylopectin, cellulose cannot be digested by humans. The reason is that we cannot synthesize the enzyme cellulase, which can hydrolyze those beta-1,4 glycosidic linkages of the cellulose polymer. Indeed, only a few animals, such as termites, cows, and goats, are able to digest cellulose. These animals have within their digestive tracts microorganisms that produce the enzyme cellulase. The sugars released by this microbial digestion can then be absorbed and used by these animals. In humans, cellulose from fruits and vegetables serves as fiber in the diet. And here's the structure of cellulose, and you can see the beta-1,4 glycosidic bond. Of course, it's beta because the bond is going up like this, and the 1,4 comes from 1 on the anomeric carbon and 4 on the next carbon of the neighboring glucose molecule.